Hello, my name is Jay Huenemann. I am a research scientist at Corteva AgroScience. My background is in bioenergy and biofuels. I obtained a degree in energy sciences and engineering from the University of Tennessee. And right now I work in ag biotech. The work we do here at Corteva is across the entire agricultural industry, but my work is focused on biologicals. That means plant and seed treatments that are derived from biology. So most of our products are derived from microbial systems. So we work with a lot of bacteria and fungal systems to isolate and engineer new herbicides, pesticides, or biostimulants. My work specifically as a research investigator is as a synthetic biologist. I work heavily in molecular biology, working to do metabolic engineering for secondary metabolites. So most microbes have evolved systems to either protect themselves or make themselves useful to plant system to enable them to live symbiotically. And so we screen large libraries of microbes for novel compounds that have one of these functionalities. And then once we have microbes that have a phenotype that we're interested in, we take those, we generally do a whole genome sequence to figure out what their DNA looks like. And then we can also do things with their RNA and protein sequences to figure out what their DNA transcription translation systems look like. And that helps us identify the pathways and genes that are involved in these secondary metabolites that are being produced. And so once we've done that, then we typically look for ways to modify or improve those systems to be either more efficient or more effective at producing higher amounts of the product. So for things that we want to put on a plant as a fungicide or something, we want to generate as much of that as possible through our fermentation processes. And so my work and my goal is to take those microbes that we find with novel phenotypes and use synthetic biology, genetic engineering tools to make them work more effectively and more efficiently through our fermentation sciences to build more robust biological products that we can sell into the agricultural market and help improve crop yields and productivity. So my role as a research investigator involves the formulation and execution of experiments to engineer microbes. What that involves is a lot of analysis of genetic information, so viewing and visualizing DNA sequences, protein sequences, using various software tools to manipulate and edit those sequences. So in the lab, what that means is I do a lot of molecular biology. This includes DNA extractions, PCR amplifications, gel electrophoresis, bacterial transformations and things like that to put DNA into the microbes that we're working with. A lot of that work can be laborious and it takes time and it takes days because biology is not always fast. And so some experiments and processes that we run through can take weeks to complete a single experiment, depending on the organism we're working with. Not all organisms are like E. coli and grow in a single day. A lot of the things we work with require very specific medias and growing conditions and can take up to weeks to grow. And so the time and effort it takes to engineer some of these microbes is pretty intense and can require a lot of focused effort. But the research is very rewarding when we are able to take one of these what we call non-model microbes, and we can engineer them to improve their efficiency or their titers and yields. And once we do that and we can move it into product development stage, that's a really rewarding part of the work, because then we start moving towards having a system that we've made that will actually give us an agricultural product, or a product that we can put into the world to help farmers do a better job at what they're doing. So that's a big piece of what drives our work here.